Hello, it's Dr. Morris, and today we're talking about immunology. This is a very brief review of a very important topic. You will be addressing some of these concepts again in the infectious disease course that will occur in a later semester. I think it's probably summer with Dr. Greenwood. But I wanted to go through some of the important concepts in immunology because this is not something that's addressed in your book, and it is something that's extremely important in terms of physiology. The immune system is a group of cells and structures that create the ability of the body of the organism to resist infection and other kinds of diseases. And there are a lot of ways we divide the immune system, so you will be hearing some fairly arbitrary divisions. One of which is that we consider some of the immune system to be cellular, and obviously that involves certain cell types like white blood cells, for instance, and other parts of the immune system we consider to be humoral, meaning that they are various chemicals, proteins, enzymes that are in the circulation or in the extracellular fluid, and that are in all cases obviously produced by cells, but that which work in isolation from cells or along with cells to help resist infection and disease. And truly, all of the cells in the body and all of the tissues in the body are involved in immunity. It's not just the lymphatic system, it's not just the blood cells that we've talked about, but immunity is a large and complex part of the way in which the organism resist infection to maintain homeostasis. So, among other things, the lymphatic system, or primarily the lymphatic system, protects us against disease. Not only infection, but primarily infection, also neoplasm or cancer. So the cells in the lymphatic system have ways of responding to things like pathogens that are in the environment, to certain toxins that can cause disease when the body's exposed to them, and even to abnormal cells in the body, such as cancerous cells. So, when we're talking about pathogens, we're talking about microscopic organisms that cause disease and a few macroscopic organisms. So we're talking about viruses, about bacteria, about fungi, and about parasites. Some parasites are microscopic, single-celled, or very small multi-celled organisms. Some parasites are macroscopic. Some worms are quite large. But each of these pathogens attacks in a specific way, and the immune system has very specific ways in which to fight them, along with some general ways. So, the role of the immune system is to defend against infections, and we use the immune system in various ways. We manipulate the immune system by things like vaccines. So we have developed ways to boost the defenses that the immune system provides to help protect against infection. The immune system helps to defend against neoplasm, against cancer, against tumors. And as we manipulate the defense against infections, we can also, in the current era, manipulate the defense against tumors. And there are increasing types of immunotherapies that have been developed that are used to defend against cancer. The immune system responds to newly introduced molecules and to tissue grafts, and that can be both an opportunity and a threat. So the immune system's responses are barriers to the successful transplantation of tissues that are not autologous, that are not from the individual. So person to person transplantation, and of course xenografts, transplants from non-human organisms to human organisms. On the other hand, we can manipulate the immune system to better allow the immune system to accept these foreign materials, and we can use gene therapies which can be either resisted by the immune system or can help us to manipulate the immune system. And then finally, the immune system, while it has a lot of positive roles, can also cause cell injury and can induce its own pathology by triggering inflammation. And this can happen during what we call allergic diseases, where the immune system is overactive in response to molecules or materials that have been introduced, and autoimmunity, where the immune system pathologically develops responses to to cells that are part of the organism or part of the self, and other inflammatory diseases in which the immune system's activation increases pathology. So while the immune system is extremely important in defending us against threats like pathogens and cancer, it's also the cause of a number of really serious diseases that have significant morbidity and mortality, and it's the major barrier to the transplantation of organs and to things like skin grafts. So I mentioned earlier that one arbitrary way to divide the immune system was into cellular and humoral. Another arbitrary way to divide the immune system into two arms is that we have innate defenses that are non-specific, and we have adaptive um, uh, mechanisms, defenses, that are very specific and have to be acquired. So the innate or non-specific defenses will work in the same way against any type of invading agent. On the other hand, the adaptive defenses protect in general against specific pathogens and are dependent upon the activities of the white blood cell type that we call lymphocytes. And in order for an adaptive defense to be enabled, the organism has to be exposed to the specific hazard or pathogen. So innate non-specific defenses are older and phylogenetically older. They've been around for much longer in terms of biology. But they always work the same way, and they work against all attackers or all comers. They are non-specific, whereas the adaptive defenses are evolved considerably later, protect against specific organisms and pathogens, depend on lymphocytes, and develop in response to exposure to organisms or hazards. So in this diagram, we are looking at a timeline showing some of the mechanisms of innate and adaptive immunity. Innate immunity, nonspecific immunity, tends to be much more rapid. And because it's nonspecific, it can go into action right away. It doesn't matter what the threat is. This innate immunity is here to attempt to handle the threat. So one important part of innate immunity is the epithelia that line the interfaces between the organism and the environment. And so those epithelia include the skin. They include the lining of the respiratory tract, the lining of the genitourinary tract, the lining of the gastrointestinal tract, and all of those are barriers that prevent the microbes on the outside from getting inside, from getting through the barrier into tissues where they can be attacked. In addition, there are cells that are phagocytic. The many white blood cells, including the polymorphonuclear leukocytes or neutrophils or granulocytes that we talked about in the blood and coag lecture, are phagocytic, and they can reach out, attach to, and engulf the organism, and using enzymes kill and break down that organism. There are cells called dendritic cells that are also phagocytic, and these cells are not just circulating in the blood, but within tissues adjacent to epithelial barriers in order to try to capture and destroy microbes that have invaded and gotten through these epithelial barriers. Epithelial barriers can be disrupted. So when you fall off your bike and scrape your knees and elbows and disrupt the epithelial barrier, there are phagocytic cells waiting to capture and destroy the organisms. There is a system called the complement system, which is a cascade of proteins that are circulating in the blood and the extracellular fluid. We'll talk a little bit more about that in a few minutes. And there are cells called natural killer cells that are nonspecific lymphocytes that will attach to and inactivate organisms. So, whoops, sorry, we're going the wrong way here. So again, if we're dividing the immune system and the immune response into these two arbitrary responses, the innate response and the adaptive response. In the innate response, microbes that manage to get through those epithelial defenses are attacked by phagocytes, by natural killer cells, and by proteins, particularly the proteins of what we call the complement system or the complement cascade. And that is a universal response. It does not matter what the organism is, any microbial organism, or in fact other breaches, including things like neoplastic cells, are going to be attacked in this way. The innate responses actually enhance the adaptive responses. So the innate responses and the adaptive responses work well together, and it's thought that the adaptive response probably evolved as certain organisms evolved to resist these innate responses. For instance, there are organisms that, when phagocytosed, resist being killed and actually take over the cell that eats them, or is able to continue to grow and multiply and actually subvert the process of the innate phagocytic response. So the adaptive response involves lymphocytes, and there are B cells and T cells. We've already talked about the natural killer cells and the innate response. T cells provide cellular immunity, and B cells make antibodies, which are part of the humoral immunity. So leukocytes is the word that we use to talk about white blood cells. Okay, white blood cells are leukocytes. Leukocyte just means white cell. And leukocytes, unlike erythrocytes or red blood cells, don't have hemoglobin and do have functional nuclei and other organelles like mitochondria and endoplasmic reticulum and Golgi apparatus. They do things, they make things. They often have vesicles that contain enzymes. And white cells function in a number of ways. They defend against pathogens, but they also tend to graze and remove damaged cells, wastes, and toxins. Things like dust may get phagocytosed by white cells. And in addition, white cells attack abnormal cells, so the cells that have a tendency potentially to go down the road to malignancy may be attacked and removed by leukocytes. And we have talked about neutrophils extensively. These neutrophils, monocytes, eosinophils, and basophils, if you remember, come from the line in the bone marrow called the myeloid line. They are myelocytic cells, whereas lymphocytes come from the lymphoid line. We can remember the types of white blood cells as never let monkeys eat bananas, neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And here we have a neutrophil. This is a lymphocyte. This is a basophil. This is an eosinophil. And this is a monocyte. Now, circulating white cells have some characteristics. They are able to leave the vessels, migrate out of the bloodstream, kind of squeeze their way between endothelial cells so that they can get out to the tissue when signals are there to attract them. They do this in a way very similar to how amoeba move. So they have amoeboid movement. They use microtubules and extend arms to attach and pull themselves. And they are attracted to specific chemical stimuli, which is a characteristic called positive chemotaxis. Many white blood cells are phagocytic, so neutrophils, eosinophils, and monocytes can all engulf and consume abnormal intruders, microbes. When we are looking at white blood cells in the laboratory, we're looking at a number of things. We're looking at the total number of white blood cells, but then we look at something called the differential, and the differential is what are the different white cells, how many are there. So in addition to having a total white blood cell count that might be in a normal person 8,000, we're also going to look at how many of that 8,000 white blood cells are neutrophils, how many are lymphocytes, and how many are those others which occur in smaller numbers. And that differential count lets us kind of see changes in the populations of white blood cells that occur in response to specific insults. So Bacterial infections will often cause a large rise in the neutrophil count. Some types of inflammation will do the same. Allergic reactions may cause an increase in the number of eosinophils. Inflammation, often back to neutrophils or granulocytes. There are some disorders of white blood cells that are defined here. Leukopenia just means not enough white blood cells. So that's an abnormally low white blood cell count. Normal white blood cell count, I think, is something like 3,500 to 9,000 or 10,000. Leukocytosis means an abnormally high white blood cell count. Leukemia is actually a clone of abnormal malignant cells that causes an extremely high white blood count. So leukemia means white blood. People with leukemia tend to not make enough red cells, and they have a huge number of white blood cells. Now, lymphocytes, which are the ones we're going to focus on here, make up about 20 to 30% of the circulating white blood cells. So the largest number of white blood cells are neutrophils or granulocytes. The next is lymphocytes. They are a little bit larger than white cells, but they're smaller than neutrophils, and they migrate in and out of the blood. So we find a lot of lymphocytes in lymphoid organs like lymph nodes and the spleen, and in connective tissue really throughout the body. And they are part of the body's innate defenses, specifically natural killer cells, and the specific defenses. And there it's primarily the T cells and the B cells. So when we're talking about major categories of nonspecific defenses, we're talking about physical barriers, epithelial linings, phagocytes, including neutrophils, monocytes, eosinophils, immunological surveillance, which there are cells which find and detect abnormal and foreign substances and present them to the immune system, and we'll talk about those a little later. Chemicals called interferons, proteins in the complement cascade, what we call the inflammatory response, which occurs when there is generally a problem which needs to recruit immunological cells, so cytokines and chemotactic agents, chemicals are released which cause white cells to come to the area and release additional substances which cause things like the capillaries to become more permeable, therefore swelling, redness, heat, and then fever. So here we have an illustration of the physical barriers that keep organisms outside of the body. Some of the kinds of phagocytes, monocytes, macrophages, neutrophils, eosinophils. Immunological surveillance. So immunological surveillance is interesting in that natural killer cells find and destroy abnormal cells. So diseased cells, malignant cells are attacked by natural killer cells and lysed. Interferons that are chemical messengers that are really important in coordinating defense against viruses. So interferons are released by lymphocytes and macrophages, but also by viral infected cells. And then this complement system, which is interesting and we'll talk about a little bit more, which consists of these circulating proteins that work together with antibodies to lyse pathogens, to break them up, break them down. the inflammatory response, and we talked a little bit about these things, these cytokines that are released by immune cells cause increased blood flow, activation of phagocytes, increased capillary permeability, activation of complement, often clotting to wall off the region, an increase in temperature locally, and activation of the adaptive defenses, so the T cells and the B cells. And fever is an innate defense that accelerates the metabolism within tissue and the activity of the defense cells. We have two classes of phagocytes, small ones and big ones. So they're called macrophages, which include neutrophils and eosinophils, and that leave the bloodstream and enter the tissues to fight infections. And then we have something called macrophages. So there are cells circulating, white cells in the blood called monocytes, but the monocytes tend to distribute themselves out of the blood throughout normal tissues, where when activated they become macrophages. So they are phagocytic. They are full of granules of assorted cytokines and other chemotactic agents that activate the immune system locally and cause inflammation. So activated macrophages can engulf a pathogen because they're phagocytes and destroy it with the enzymes in their lysosomes. They can bind to a pathogen so that other cells can come and know to destroy it. And they can actually release some of their lysosomes and granules, the contents of their granules, into the interstitial fluid to act on the pathogen and cause destruction. Coming to complement, there are 11 complement proteins. I remember having to memorize the complement cascades in medical school. I don't think it's terribly useful, but I do think it's important to know a couple of things. First is that there are complement proteins, and there are a bunch of them. So we call those C proteins or complement proteins. They work together, acting on each other in what we call cascades, similar to the way that the intrinsic side of the clotting cascade or the common pathway, the clotting cascade works, where one acts upon another, which acts upon another, which results in some specific final event that causes cell death. And there are two pathways. The classical pathway and the alternative pathway. So in the classical pathway, basically, complement 1 binds to antibody that's attached to antigen. So say you have a bacteria, say you have a strep bacteria, and say you have antibodies that have been made by B cell lymphocytes to the strep bacteria. So they've bound to the antigen in the wall of the bacteria. The C1 binds to that antibody and then acts as an enzyme to start a chain reaction that essentially results in the attachment of additional reactions of additional complement proteins. So this is considered to be the classical pathway. It's fast. So we have antibodies attached to the bacterial cell wall. C1 attaches, activating this cascade that involves additional complement proteins. One of those complement proteins attaches to the cell wall and basically punches a hole in it and causes cell death. Okay, the other alternative pathway is considered a slow method. It involves other proteins, other factors, but again, it results in activated C3 attaching to the cell wall, which punches a hole in it. And that hole basically is called the membrane attack complex, and makes the protein lyse by fluids rushing in and bursting it. In addition, it enhances phagocytosis. So, we've talked a good deal about innate immunity, which is the phagocytic cells, the barriers, the complement pathway. And now we're going to talk about natural killer cells. Now we're going to talk about adaptive immunity. And here again, we're making our two arbitrary divisions and discussing humoral immunity and cell-mediated immunity. So B lymphocytes produce antibodies, and antibodies mediate this part of the immune system we call humoral immunity, because antibodies are circulating in blood and fluid, and therefore humor. The humor is the fluid. So humoral immunity involves antibodies that are produced by B lymphocytes. Humoral immunity works against microbes that are extracellular, that are reproducing outside of cells. Antibodies cannot get to microbes that are reproducing and living within cells. So humoral immunity only works against certain specific organisms, but we already knew that because this is a type of adaptive immunity, so it is specific. Cellular immunity that is mediated by T cells, including cytotoxic T cells and helper T cells, so killer cells and helper cells, tends to help us defend against microbes that live and reproduce within cells. So this is a defense against intracellular microbes. Most T cells recognize only antigens that are proteins, whereas B cells, and therefore the antibodies that they make, can recognize other types of molecules. So proteins, carbohydrates, proteoglycans, nucleic acids, and lipids. So we can have antibodies against many different kinds of molecules, but T cells only are able to recognize, to detect, and to respond to protein antigens. So here we have a diagram of adaptive immunity showing on the left side humoral immunity, where we have extracellular microbes, bacteria. And I'd like you to think about these bacteria. We have both cocci and rods, right? And these cocci are single and in pairs. This is a short chain of rods. Just saying, we've talked about this, or we will talk about this. But when a B lymphocyte responds to these bacteria, it learns how to make antibodies to it. And those antibodies are secreted from the B cells, attach to the bacteria, and then help to neutralize the bacteria, may activate the complement system to lead to lysis of the bacteria, or may assist in phagocytic cells in opsonizing or eating those bacteria. On the cellular side, we have intracellular microbes, in this case viruses, but there are some intracellular bacteria as well. This is a little bit more complex in that the antigen is presented to the T cell system, to a helper T cell, by an antigen presenting cell. So the bug has gotten into the cell, it is processed, it is presented, and then that T cell to which it's presented causes cytokines to be produced that causes proliferation of additional T cells that are going to act against that specific microbe. And so we have activation of effector cells that include cytotoxic T cells and macrophages. They are going to lyse the infected cells, which will be cleaned up by the phagocytosis, by the phagocytes. So in both cases, phagocytic cells are going to be involved. The T cells are called T cells because they are thymus dependent, so they are processed in the thymus. B cells are bone marrow derived, so that's where the names come from. And then the third kind of lymphocytes are natural killer cells, which are involved in innate immunity, but also become active in adapted immunity as specific organisms are being attacked. So here we have a common lymphoid precursor. You remember we talked about this when we talked about blood and bone marrow. And that lymphoid precursor is in the bone marrow. It can divide into B cells or T cells. The B cells remain in the bone marrow until they are mature. The T cells go to the thymus where they mature. Immature B lymphocytes get released into the circulation where they mature, and the T cells that have been processed mature in the thymus and get released into the bloodstream. These T cells and B cells can either stay in the bloodstream or they can leave it and enter peripheral lymphoid organs that include the lymph nodes and the spleen, other lymphoid tissue, things like the tonsils and the adenoids are lymphoid tissue. So there are lymphoid tissues on mucosa and near the skin. And these cells are kind of recirculating in and out of the blood and in and out of the peripheral lymphoid organs. About 80% of circulating lymphocytes are T cells. So T cells can further differentiate. There are a number of kinds of T cells. There are cells that are called cytotoxic cells, and they attack foreign cells or body cells that are infected by viruses. They're cytotoxic. There are helper T cells, and helper T cells activate both T cells and B cells, so they're involved in the adaptive immunity of both T and B cells. Suppressor cells inhibit activation of both T cells and B cells. And memory T cells are a group of these T cells that have responded to an antigen. They've encountered an antigen, they've responded to that antigen, and now they stay in circulation providing memory for that antigen, and they can reactivate when that antigen comes back. B cells make about 10 to 15% of circulating lymphocytes. So they're B cells that are circulating. Those circulating B cells, when necessary, with the help of a T cell, of a helper T cell, become a plasma cell. And plasma cells are little factories for making antibodies. They make immunoglobulin proteins. So we talked about immunoglobulins, globulins as being part of the circulating proteins in the blood, lower quantities than albumin, albumin made in the liver, immunoglobulins made by circulating plasma cells, and plasma cells in lymphoid tissue. When a B cell is activated, and antibodies are required, it differentiates into one of two kinds of cells: plasma cells, which are the factories for making antibodies and synthesize and secrete antibodies, and memory B cells. And just like memory T cells, the memory B cells stay in circulation and are part of how we respond the next time the same antigen is encountered. So you have a strep infection. Some of your B cells get activated by helper T cells and differentiate into plasma cells that start making antibodies against strep. But some of those B cells divide into become memory cells, and those memory cells continue to circulate. Your immune system takes care of the strep with or without the help of antibiotics. Strep is usually a self-limited disease. And ultimately, the next time you're exposed to strep, they are ready to jump into action, become activated, make more plasma cells and more memory cells. So when an antigen or a foreign molecule triggers the immune system, and antibodies or immunoglobulins are made, immunoglobulins are specific. They are specific to the organism that has triggered the response. Immunoglobulins are interesting proteins. They're made up of what's called heavy chains and light chains. And when the specific antibody encounters its target, which is the specific antigen on the bacteria, it initiates something called antibody-mediated immunity, which is this chain of events that destroys the organism. So it can do one of three things. When enough antibody attaches to a bacteria or a virus, it basically inactivates that pathogen by keeping it from being able to attach to anything, so to the target tissues. So the antibodies can just neutralize the bugs. It can promote phagocytosis. It can help the local phagocytic cells, the neutrophils and the macrophages, to eat it up. And it can activate complement, which results in lysis of the cell and also enhances opsonization. So that's humoral immunity that we've talked about. We have the microbes, we have lymphocytes responding. Lymphocytes turns to a B lymphocyte, which secretes up into a plasma cell, which secretes antibody. The antibody either blocks or increases the infection and eliminates the microbes. On the cellular side, things happen a little bit differently. So there are kind of two possible ways this is going. The macrophage has found and ingested a microbe, so they are intracellular. Or the microbe has infected a cell, so they are intracellular. So here we have intracellular organisms. The T lymphocytes, in the case of macrophages, helper T lymphocytes work with the macrophage to eliminate the microbes that have been phagocytosed. These are microbes that can live within the cell. So they've been phagocytosed, but they're still alive. With the help of the T lymphocyte we are able to get rid of those cells. In the case of the intracellular microbes in infected cells, cytotoxic lymphocytes will kill the infected cells. So they're not just killing the microbes, they're killing the microbes and the infected cell. I'm going to talk for a little while about a phenomenon called hypersensitivity, and we'll be coming back to T cells because there are a few different kinds of hypersensitivity. Hypersensitivity type 1 is often what we call allergy, although other kinds of hypersensitivity are sometimes called allergic disorder. But basically, type 1 hypersensitivity is what we typically think of as allergy that causes things like food allergies, hay fever, asthma, and eczema or atopic dermatitis. Type 2 hypersensitivity is a little bit different. We'll come back to that. Type 3 hypersensitivity and type 4 hypersensitivity cause reactions that are quite different to the rapid reaction of the type 1 hypersensitivity. Normally, the tissues of the body are the immune system is tolerant to self. It doesn't respond to self-antigens. And this starts during fetal life, that as lymphocytes are formed and many different kinds of receptors are formed on the lymphocytes, you'll produce lymphocytes that can recognize antigens that are self and don't respond immunologically to these self-antigens. So we can kind of live with ourselves under normal circumstances without attacking our own cells. In the cases where the mechanisms to prevent immune responses to these organisms fail, we develop immunity to our own cells, and we can have what's called autoimmunity. This can be either cellular or humoral. If we are making antibodies to our own tissue, that's humoral. If we have T cells attacking, then that is cellular. When we have breakdowns in our tolerance to antigens, we develop either hypersensitivity or autoimmunity. Hypersensitivity to foreign substances, autoimmunity to self substances. So hypersensitivity is an immune reaction that causes damage, and autoimmunity are responses against our own self-antigens. And in either case, this breakdown in tolerance to specific antigens, either things that are commonly in the environment or our self-antigens, can result from either our genetics, so our genetic makeup, specifically in regard to the human lymphocyte antigen genes called HLA alleles that are part of how we identify HLA alleles. HLA types are used to identify potential matches for transplants, to identify closer relationships between tissues and organisms. But you can also get breakdowns in tolerance from environmental exposures. So for instance, from infections that mimic self-organisms. So certain strains of strep, for instance, result in the immune system making antibodies that can attack heart valves, resulting in rheumatic fever, or that can attack the kidneys, resulting in post-streptococcal glomerulonephritis. So there are a few different types of hypersensitivity disorders. As I said before, type 1 is what we generally call allergy and results in things like allergic rhinitis or hay fever, asthma, eczema, atopy, which is a term you've encountered in dermatology. And type 1 hypersensitivity disorders are mediated by IgE. Type 2 hypersensitivity disorders refer to autoimmunity in which an antibody is produced that directly acts on a self tissue or receptor. An example of that might be something like rheumatoid arthritis, in which antibodies are made that act on the lining of the joints, resulting in inflammation. In type 3 hypersensitivity, the antigens that are circulating combine with circulating antibody, creating complexes that activate complement, causing tissue destruction, self-tissue destruction. And type 4 hypersensitivity is also called delayed hypersensitivity and is completely independent of antibodies, is only cell mediated. So let's talk briefly about allergy. These are some diagrams from Nature Reviews on Immunology that are showing basically IgE, which has been produced by a T cell. It's an immunoglobulin, so it's produced by a B cell, sorry, not a T cell, as a result of a dendritic cell, which is a cell sitting in the under the mucous membranes, for instance, or in the skin, that has contacted a antigen in the environment that we'll call the allergen. And that allergen has been picked up by IgE on the surface of the dendritic cell and is presented to a T cell. And that T cell interacts with a B cell, and you don't have to know these details. That B cell then ends up basically differentiating into a plasma cell that's going to make a lot of antibody to this allergen. And that antibody is immunoglobulin E, IgE, which is now circulating and resting and hanging around. When that allergen is encountered again, particularly to IgE that has attached to the surface of a kind of immune cell in the mucous membranes or the skin called a mast cell. That antigen attaching to two separate IgEs and linking them causes the mast cell to release a lot of chemicals, including histamine, which is a chemical that causes blood vessels to become dilated and leaky, cytokines, which are going to be recruiting additional immune responses, leukotrienes, prostaglandins, all of these participate in an inflammatory process that causes swelling and leaking of tissue, and therefore a stuffy, runny nose, for instance. So IgE is really important in the activation of allergic inflammation. IgE that has been made by plasma cells is attached to the surface of mast cells when antigens, and this is perhaps showing pollen, cross-links two of these IgEs on the surface of the mast cell, causes an immediate release of a number of substances, including the histamines, and then tumor necrosis factor alpha, some proteases, heparin. Over minutes, prostaglandins and leukotrienes, and over hours, cytokines, including the interleukins. So the allergic inflammation has both an immediate response and a delayed response. Okay. Histamine works fast. So it happens really very rapidly after the mast cells are degranulated, causing vascular congestion and edema and recruitment of additional cells, including eosinophils. The early reaction is the mast cell degranulation. The later reaction comes from these other products, the prostaglandins, leukotrienes, and especially the cytokines, which are recruiting other immune cells to the area and increasing the inflammation. So we have an immediate reaction and a late reaction. So this is kind of important. When you see somebody, for instance, in the ER who has a bee sting allergy, they have been stung by a bee, that antigen in the bee venom has caused lots of mast cells to degranulate, probably in the skin causing hives, but also in the respiratory system, potentially causing edema that's going to cause bronchospasm and make the airways close up. And that's a rapid response, and we can treat it with things like adrenaline, and antihistamines potentially, and an hour later that person might be better. But if we do nothing else, the late phase reaction will mean that some hours later all the inflammation is going to come back. And in fact, this is why when we see those people initially, we also give them steroids to damp down this late phase reaction, kind of turn off the immune system. And so 4 to 6 hours after the initial reaction, we're going to have much more inflammation unless we do something to interrupt it. So there are a few different kinds of IgE-mediated reactions. Itching and dripping of the nose, in the skin, urticaria or hives, in the bronchi, edema of the wall of the bronchi, along with contraction of the smooth muscle and bronchial constriction. And then other things like potentially large bronchi constriction, laryngeal edema causing stridor, potentially hypotension because of diffuse release of substances that cause vasodilation. In the gastrointestinal gastrointestinal system, nausea, vomiting, and abdominal cramping are common responses to IgE-mediated reactions, particularly in food allergies. So food allergies are not all that common. Among the ones that occur quite commonly in children are milk and egg and peanuts. So there's about a 5% food allergy rate in infants and young children. In adults, there's about a 3 to 4% range, but the most common food allergy in adults is to shellfish. But parents think their kids have food allergies. So up to a third of parents think their kids have food allergies. This is more objective data based on widespread allergy testing. On the other hand, allergic rhinitis is very common. So up to 40% of children and maybe 20% of adults tend to have runny noses during seasonal rhinitis or perennial rhinitis that's a result of IgE-mediated allergy, type 1 hypersensitivity. And here we have a few things. We have pollens that cause those reactions. We have fungal spores, and we have critters. This is a dust mite, which is a common cause of allergic rhinitis. Asthma is an IgE-mediated atopic hypersensitivity, type 1 hypersensitivity reaction that is inflammatory. As a result of the IgE-mediated mast cell degranulation, we get spasm of the bronchi, sputum phlegm production, and swelling that causes bronchoconstriction. Anaphylaxis is a dramatic form of IgE-mediated allergic reactions, and it's not just hives. Hives by themselves are not anaphylaxis. Typically, anaphylaxis occurs within a fairly short time, but it's not always immediate after exposure. So say somebody has a peanut allergy and they eat some peanuts. Well, within a relatively short time, up to 2 hours after the exposure, there is going to be a massive release of histamines and the other substances like prostaglandins and leukotrienes from mast cells and basophils that are going to cause all of these things: hives and flushing, constriction of the airways, and that can be either upper, so like laryngospasm, difficulty speaking, stridor, lower airways, bronchoconstriction and wheezing, potentially abdominal cramps, vomiting, diarrhea, and lightheadedness, which is because of diffuse vasodilation, you get a drop in the blood pressure. And so anybody with anaphylaxis or with potential anaphylaxis with hives who's complaining of feeling lightheaded, you should get a blood pressure immediately, and then potentially palpitations. People developed anaphylaxis, a common source of anaphylaxis, 1% in children, are peanuts, peanut antigens, peanut proteins. The proteins that are associated with latex, these blue gloves are probably nitrile, but latex gloves are pretty common. Food is the main cause of anaphylactic cases in emergency departments. Latex can cause anaphylaxis. Obviously, insect stings can cause anaphylaxis. So to diagnose allergic disorders, history is really important. Physical exam is important. But when it is suspected, it is worth getting skin testing. So skin prick testing, which is generally done by allergists and in some communities by ear, nose, and throat doctors, can test for environmental allergens and also for food allergies when they're suspected. When you get a negative result, you can be pretty sure that it's accurate. Positive results sometimes are fairly nonspecific. So some skin prick testing is overly sensitive and insufficiently specific. When there is concern, you can also do IgE testing, which is called Immunicap IgE testing. This is a blood test, so it does not involve skin prick testing. And it also has a high sensitivity and a relatively low specificity. So the negative predictive value is high and the positive predictive value is lower. This is a picture of a skin prick test. Each of these little sharps is impregnated with a specific allergen so that we can kind of penetrate the epidermis. A little bit of the antigen is injected into the dermis, and we number to look for what responses we get. There are usually controls which always cause a positive response. And this is a generally positive skin prick test. So the only negative is 2, and I wouldn't be surprised if 2 was a negative control. Okay. Immunicap testing is a blood test for specific IgE that uses anti-IgE for specific antigens and measures the fluorescence. If there's very little IgE, you get a low level. Anything above indicates sensitization, but does not necessarily mean that the person is clinically allergic. So this has to be used along with potentially skin tests and history. Just because somebody has IgE against, for instance, peanut antigen or milk antigen does not necessarily mean that clinically they are allergic. But if they have a reaction when they eat milk or drink milk, and then respond with a positive have a positive Immunicap IgE result, that's going to suggest allergy. And then finally, this is somebody with asthma getting a pulmonary function test. We see our loop here, including our various measures. FEV1 is the forced expiratory volume in 1 second. These are a combination of volumes and velocities for inhalation and exhalation, so flow. In people with asthma, we see what's called an obstructive pattern. So they are able to inhale normally, their exhalation is slowed down. And we can give albuterol before and after to see whether that the person has reversible airway constriction. In addition, you can provoke bronchospasm by giving either methacholine, which is an acetylcholine agonist, or for people who have exercise-induced asthma, doing an exercise challenge. Chest x-ray is usually part of the diagnostic profile for asthma because you want to rule out other potential conditions that would show up on chest x-ray. Asthma does not particularly, except for sometimes hyperinflation in people during acute exacerbations. How do we treat allergy? Well, with food allergy, the best thing to do is simple avoidance of the suspect food. With allergic rhinitis, environmental controls are really important. So getting rid of that cat, if the cat is what's causing the allergy, may be helpful. Spending time in indoors, air-conditioned environments during times when there are high levels of the pollens that we're allergic to. And then topical nasal steroids, and potentially antihistamines. And allergy shots, which are very effective in a lot of people in desensitizing and decreasing the reaction to the allergen. Asthma is treated in a couple ways. When there is an acute exacerbation, we use a short-acting beta agonist as a rescue treatment, just to relax the bronchial smooth muscle. But it's also very helpful to control the inflammation because asthma is an inflammatory disease. It is not only smooth muscle constriction that is causing the bronchi to be constricted. It is also edema. So we want to control exposure to the antigens Typically for people who have moderate to severe asthma, we use inhaled steroids with or without a long-acting beta agonist. Singulair is interesting because it is an antagonist to leukotriene receptors. So the leukotrienes that are released by mast cell degranulation are unable to attach to the receptors and cause further inflammation. There are agents that are specifically anti-IgE, and theophylline clinically is no longer or rarely used anymore. With type 2 hypersensitivity, there is antibody against a tissue antigen, and that antibody can target the tissue directly. Like I mentioned, in rheumatic fever, it's through molecular membrane. In Goodpasture syndrome, which is a kidney disease, the antibody is to the basement membrane protein. But in addition, complement can get activated, causing inflammation and tissue injury. The second mode of tissue injury in type 2 hypersensitivity is that the circulating cells get coated with antibodies and end up lysing the cell, which gets phagocytosed. And hemolytic disease of the newborn, which is Rh disease, is because of antibodies to membrane proteins on the erythrocytes of the child. And in that case, the cells lyse, creating hemolysis. The third mode of inducing tissue injury is by activating or inhibiting a tissue receptor. So in Graves' disease, if you remember, there are antibodies made that act as though they're TSH, and they attach to the TSH receptor and don't let go, causing constant stimulation. In myasthenia gravis, the antibodies block the acetylcholine receptors, not allowing, they attach to and block the action of the acetylcholine reactors, not allowing the muscle cell to depolarize. Treatment for type 2 reactions involves, ideally for hemolytic disease of the newborn, prevention. So when a mother is Rh negative, anytime she is pregnant, we need to give her RhoGAM, and anytime she has a miscarriage or a delivery, we need to give her RhoGAM, which is an antibody that binds up the Rh antigens so that the antigen isn't recognized, so that the immune system isn't sensitized and does not start making the anti-Rh antigens. We can also give immune suppression. So we can suppress the immunity by giving a lot of IVIG to suppress production of autoantibodies, and we can give steroids to basically turn off the immune system. The classic example of type 3 hypersensitivity is systemic lupus, in which antibodies bind with antigen and become what are called circulating immune complexes. And those immune complexes then can get deposited within vessel walls, causing vasculitis or inflammation of the vessel. This can happen in the kidneys, in the skin, in the joints. And those antigens may be either foreign antigens or self-antigens. When the antigen-antibody complexes are deposited, which is usually in the walls of small vessels, then the whole complement cascade is started, and you get inflammation as a result of that. And the pathology is the result of where that complex is deposited. This is just a picture of vasculitis, in which immune complexes have deposited in the small vessels of the skin, causing complement activation and cell destruction, and consuming complement and resulting in low serum complement levels. Serum sickness is a type 3 reaction. The classic serum sickness used to occur in response to people getting horse sera, so horse antibodies to treat illnesses before we had the availability of antibiotics. And symptoms begin within a week after the first exposure to the agent, and these days sometimes occurs in response to antibiotics, resulting in rashes, fever, and joint pain. The antibodies initially are IgM, become IgG, and this goes away if you take away the triggering antigen. But it happens more quickly if that drug is given again. We treat this with antihistamines, treating the fever, systemic steroids, and in some cases, certain other agents. And we can also remove circulating immune complexes by removing plasma, so plasmapheresis, and potentially replacing it. Type 4 hypersensitivity, the classic example is poison ivy. An antigen is presented to an antigen presenting cell, say in the skin with poison ivy. So the urushiol, the lipid in the poison ivy is presented by the antigen presenting cell to a CD4 T cell. The CD4 T cell and the CD8 T cell make cytokines that result in inflammation. There are memory cells, and the next time there's exposure, those memory cells are going to recruit new cells, which are going to cause tissue injury, cell lysis, and in the case of poison ivy, the rash. And there are no antibodies at all, but there's a good deal of inflammation. This is also the result of or the cause of type 1 diabetes, where the pancreatic islet cells are destroyed. It is the result of a positive TB skin test, contact dermatitis, which is poison ivy, and is thought to be the cause of multiple sclerosis, in which myelin proteins are the proteins that the T cells are responding to. So here we show the poison ivy plant. These haptens penetrate the epidermis. They're processed by Langerhans cells. They travel to the lymph nodes, where they activate effector T cells. The cells clone, circulate through the body. Things other than poison ivy and poison oak that cause contact dermatitis include nickel, many topical medicines, particularly neomycin, some cosmetics, and some dyes, some nail polishes, and certain resins, epoxy resins in particular. And treatment for type 4 reactions includes avoidance of the antigen, steroids, either topical steroids or systemic steroids, certain other immune suppressives when the reaction is severe, and then antihistamines for itching. These are skin tests for contact antigens. So these are not pinpricks, but rather simply substances that are applied to the skin. And here we see positive reactions. That's called